enhancement, why improvement in crop production is necessary. उसके बाद फिर हमने देखा what is the meaning of plant breeding, why plant breeding technique is used, what is the purpose, for what purpose it is used. Plant breeding, उसके बाद फिर हमने देखा what are the steps in plant breeding, right? These are the steps which we have discussed, the different steps in plant breeding. And this is a flow chart which tells you everything about the steps, how this one by one steps are followed to undertake plant breeding. Then we studied the different hybrid crops which are developed in India, that is different wheat varieties and rice varieties. I told you to remember their names. Okay, this is asked for 1 mark. Apart from this, we saw a second crop, that is sugarcane. How by using plant breeding the improved variety of sugarcane is made, that we saw, right? Then millets के हम बारे में हमने देखा, how millets are developed, okay, by using plant breeding technique, how high yielding varieties of millets are developed. And there is a table which we have seen last time. ये table हमने देखा था. I told you to remember this table. This is plant breeding for disease resistance. So here the purpose is to create disease free plants, right? So which are the different diseases which affect the plants? So these are the diseases caused by fungi, bacteria and virus. Remember these names. And what type of crops improved varieties are developed to fight against, to develop resistance against this resistance against this diseases. So that is, these are the crops. Okay, these crops are developed. Wheat variety and ये जो है name है variety का, crop is wheat and variety. So you should remember this, developed improved varieties of this particular crops, right? After this, then we studied about mutation breeding, right? Plant breeding for developing disease resistance. ठीक है? Plant breeding for developing disease resistance. Under that, we studied two techniques. One is conventional technique, conventional plant breeding technique, that is the same as the steps of plant breeding. And second technique is mutation breeding. What are mutations? That we have seen last time. Mutation breeding technique, how it is used, okay, to develop plants in plant breeding, right? Up to this we saw. Then we saw this tables. This table shows you the different types of varieties which are developed to get resistance against insect pest. Okay, these are the different insect pest. These varieties are developed varieties, improved varieties of this particular crops. Okay, फिर हमने start किया था tissue culture technique, right? Last time we saw some introductory part on tissue culture. What is tissue culture technique? I explained you what is the meaning of aseptic conditions, right? aseptic conditions क्या होती है, टिश्यू कल्चर टेक्निक कैसे यूज होती है, क्या क्या चीजें, क्या क्या प्रिकॉशंस हमें लेने पड़ते हैं, वो सब कुछ मैंने आपको बताया था लास्ट टाइम, राइट? देन वी वर अप टू दिस टोटिपोटेंसी. Totipotency, I explained you what is totipotency. It is the ability of any plant cell, plant organ or a plant tissue to develop, to grow, to divide and redivide and give rise to a whole plant. It means I told you that plant में अगर कोई branch हम cut कर देते हैं, so next time after some days a new branch can develop at that particular same point on the plant. It means plant has the ability of totipotency to develop new organs from initial some part. Okay, one cell, single plant cell has the ability to grow and divide into a complete plant. This ability is not shown by animals. This is a specific characteristic of plants only, right? So as in plants, totipotency is present and hence therefore we can perform tissue culture in plants. Therefore tissue culture is possible in plants. Now this name you have to remember, Haberland. Haberland is known as the father of tissue culture. Haberland को क्या बोला जाता है? He is known as the father of tissue culture because he for the first time in the year 1902 डेवलप दिस आइडिया ऑफ इन विट्रो सेल कल्चर. इन विट्रो का मतलब क्या होता है? इन विट्रो मीन्स in laboratory, right? So he for the first time developed the technique of plant culture, tissue culture, hence he is known as the father of plant tissue culture. Okay, now the plant tissue culture medium contains, now I told you last time itself ki how we grow plants in tissue culture. It is a technique by which the plants are grown you in some specific nutrient medium, right? So nutrient medium क्या होता है? I explained you last time. By using agar we can make a solid nutrient medium, or if we want to make a liquid medium, so that also we can make. We will not use agar, a solidifying agent. So when we don't use agar, we get a liquid tissue culture medium, nutrient medium. So न्यूट्रिएंट मीडियम में क्या क्या होता है? जो भी सारे न्यूट्रिएंट्स जरूरी है प्लांट्स की ग्रोथ के लिए, वो न्यूट्रिएंट्स ऐड किए जाते हैं, राइट? सो नाउअडेज व्हेन यू परफॉर्म टिश्यू कल्चर यू गेट अ रेडीमेड पैकेट्स. आपको रेडीमेड पैक्स मिलते हैं जिसके अंदर ये सारे न्यूट्रिएंट्स जो है वो कुछ स्पेसिफिक क्वांटिटी में, अमाउंट में मिक्स किए जाते हैं. सो यू गेट अ रेडीमेड पाउडर एंड यू जस्ट हैव टू मिक्स दैट पाउडर इन some amount of डिस्टिल्ड वाटर यूज करते हैं. यहाँ पे हम डायरेक्टली टैप वाटर नहीं यूज कर सकते टिश्यू कल्चर में, बिकॉज द टैप वाटर कंटेन्स डिफरेंट काइंड्स ऑफ माइक्रोब्स. So we don't want any kind of contamination in tissue culture, hence we always use distilled water. You know the process of distillation, पता है आपको distillation में क्या होता है? What is distillation? जो भी water हम boil करते हैं, right? Water boil होने के बाद vaporization होता है. The water vapors are then cooled down and again they are collected. So that water is nothing but distilled water, which is free from any kind of microbes. So for making nutrient medium, we should always use distilled water and not simply tap water. ओके सो वो जो पाउडर होगी ये सारे न्यूट्रिएंट्स होते हैं उसमें. कौन से न्यूट्रिएंट्स है? सोर्सेस ऑफ कार्बोहाइड्रेट, सुक्रोज होगा, मिनरल्स, विटामिन्स, फैट्स. जैसे हमें ग्रो होने के लिए न्यूट्रिएंट्स जरूरी है, इसेंशियल है, वैसे ही प्लांट की ग्रोथ होने के लिए ये सारे जो भी नरिशमेंट न्यूट्रिएंट्स जरूरी है वो सारे मिला के हम वाटर में if we add to this agar, then it will form a solid nutrient medium. If we don't use agar, it will remain as a liquid itself. Okay, so likewise we can make two mediums, a solid medium or a liquid medium. Liquid medium को बोला जाता है suspension culture. Suspension means liquid medium. ठीक है? Now for tissue culture, for carrying out the process of tissue culture, a very famous medium is used. ये ready-made medium मिलता है हमें, बस उसे dissolve करना होता है water में, and that medium is called MS medium. Remember the name, Murashige and Skoog medium. These are the name of two scientists, okay, who have developed this kind of nutrient medium. ठीक है? Murashige and Skoog medium, MS medium, is usually normally used to carry out tissue culture medium. इसके अलावा भी कोई mediums होते हैं जो कि specific उस particular plant के लिए use किए जा सकते हैं. ठीक है? But this is a general medium, MS medium. Okay, now what are the other things which we should understand regarding tissue culture? So let us see that. फर्स्ट चीज तो आप समझ गए कि देयर इज अ रिक्वायरमेंट ऑफ ओसेप्टिक कंडीशन. सेकेंड, देयर इज अ रिक्वायरमेंट ऑफ न्यूट्रिएंट मीडियम, बिकॉज विदाउट न्यूट्रिएंट्स, विदाउट गिविंग एनी नरिशमेंट, द प्लांट्स दे आर नॉट बी एबल टू ग्रो. और ये हम प्लांट्स कहाँ ग्रो करने वाले हैं? लैबोरेटरी में, राइट? सो वी हैव टू गिव देम लाइट, वी हैव टू गिव देम इसेंशियल क्वांटिटी ऑफ मॉइस्चर, we have to give them nutrients. काफी उनकी केयर लेनी पड़ती है, जैसे कि हम छोटे बच्चों को संभालते हैं, a small newborn baby. Similarly, this plantlets, we have to look after them like a small kid. Okay, based on the nature of explant. Now what is a explant? Explant क्या होता है? See given here, the part of a plant used in tissue culture. So as I told you, in tissue culture, if I take any small leaf cutting, मैंने leaf, एक plant का leaf लिया, उस plant के leaf को छोटा, उसमें से बस एक छोटा square मैंने cut कर दिया leaf Okay, and then I'll use that small leaf part to grow a complete new plant. Are you getting what I'm saying? एक छोटे लीफ के टुकड़े से मैं पूरा प्लांट लैबोरेटरी में डेवलप कर सकती हूँ. So that small part which I'll use, that part is called explant. एक्सप्लांट. ठीक है, क्योंकि अगर आपको न्यू प्लांट ग्रो करना है तो देर इज सम रिक्वायरमेंट कि आपके पास कोई कुछ तो प्लांट का सेल होना चाहिए, या किसी प्लांट का कोई ऑर्गन होना चाहिए, टिश्यू होना चाहिए, राइट? सो आई विल टेक द लीफ. फॉर एग्जाम्पल, इफ आई टेक अ लीफ पार्ट, स्मॉल पार्ट ऑफ लीफ, देन it is called as the explant. Explant can be stem, explant can be leaf, explant can be any tissue, explant can be एनी सेल ऑल्सो, प्लांट सेल स्मॉल. ठीक है, तो ये सारी चीजें जो कि हमें जरूरी है प्लांट टिश्यू कल्चर इनिशिएट करने के लिए. सो दैट इज नथिंग बट द part of the plant is called tissue explant, which is used in tissue culture. Okay, so based on the nature of explant, tissue cultures can be of this types. There are three types. Cell culture. So if I want to undergo cell culture, मुझे सिर्फ cells ही culture करने हैं, right? If I take a plant cell and I start growing in it in laboratory, then it is called cell culture. If I want to develop some kind of organs, plant organs, so that type of tissue culture is called organ culture. And third is embryo culture, जिसमें कि जैसे humans का embryo होता है, similarly plants they also show embryo. After fertilization we studied zygote then forms embryo. So for this specific purposes we can also undergo टिश्यू कल्चर. नाउ बेस्ड ऑन आवर पर्पस कि हमें पूरा प्लांट चाहिए या फिर कोई स्पेसिफिक पार्ट ही चाहिए, या फिर हमें कुछ स्पेसिफिक टिश्यू चाहिए या सिर्फ प्लांट सेल चाहिए, accordingly we can do the tissue culture. Next is based on the type of in vitro growth. Okay, in vitro is always in laboratory. इसका contrast, इसका opposite word क्या होगा? In vivo. Vivo means in nature, naturally. Okay, but tissue culture is a laboratory technique. We humans are interfering, we are performing, we are helping, we are developing plants in laboratory. So it is a in vitro technique. Based on the type of in vitro growth, there are two types: callus culture and suspension culture. This